like a word bank kind of thing and go over that DNA, RNA processing again. Okay? You've seen it once on the slides now, but let's sort of get the concept more solidified in your head. And I'll write up some words here that you should know the definitions of, or at least process a sentence or two to sort of explain what's going on in your head. Okay, because that's what I'm going to try to hit at on the test. So let's just do that DNA part again. First of all, you've got that coil of DNA. Okay, it wraps twice around something, a protein here.
Histone.
Okay, these are called histones.
Right. And that forms a nucleosome.
Right, right. And when this whole process, when the DNA is added to it, then it's also called a nucleosome. Okay, so have those terms down, you understand. The importance of this is really related to which genes get turned on and off. Okay, because your body can actually, the cell can actually shut down your DNA or shut down certain genes when that particular part of the gene is wrapped around the histones. It can't be read. Okay, it has to be unwound out here so that the RNA polymerases can read it and make proteins eventually. Okay, so we can stop that process sort of artificially, and your body does it all the time. The cells will, very often they'll shut down half of your DNA, like they'll shut down your mother's chromosomes in one cell and your father's chromosomes in another cell, and only use half the chromosomes to run the cell, because most of the chromosomes have everything you need on one. Okay, remember, it's just a sort of a duplicate copy of the other chromosome. So, and the term that we talked about for using, the histones to change DNA expression, that was epigenetics. Okay, epigenetics. I'll ask you one question about that. All right. The first thing, DNA, replication, okay. Important term, that's when you've got your DNA, and let's just do it like this here. All right, you've got your base pairs. Every 3 bases is called a
triplet.
Triplet.
But it's a codon and RNA.
Right. Okay. So that's a term that you should be familiar with. And that's for coding for the proteins. Okay. But DNA replication is not about coding for proteins. It's just about copying the DNA. Okay. So you're going to get on the end of the DNA. If you want to copy the whole thing, you got to start at the end, and you have the DNA polymerase. Polymerase. So it's an enzyme that makes a polymer, basically, polymerase, and that is signaled to do that once in the lifetime of that cell, because you only need one copy, because the only thing it's going to use that for is to divide it, and so the new cell gets half of everything the old cell had. Okay, so that's what replication is for mitosis, basically, for cell division. I'll just put that in parentheses. Okay, and how it's going to do that is by splitting open the DNA. Remember, it's only hydrogen bonding that's holding it here, it's just together. And you remember from that video, you had that helicase enzyme, that blue one that was running down the DNA, separating it into 2 strands. One strand can be copied in one direction, but because the phosphate ribose bonds are in the opposite direction on the other strand, it has to be copied backwards. And that's why you get that weird flip around that the body has to do it that way. That's kind of technical, though. Don't worry about that sort of thing. Just be aware that DNA replication is when we're copying our DNA, and it's DNA polymerase that does it, and it does it only one time. It makes one copy of the DNA. And from the old test, you remember what phase that was in?
Phase. Interphase.
Interphase, right, before you go into the mitotic phase. So, all right, and that's all going to happen, in the nucleus, obviously. All right. And so we're still in the nucleus now when we want to make a protein. Or draw it like this. Okay, we have, well, first I'll do that in two phases. Let me raise it up. Let's just say here we have a strand of DNA, okay, and here's all our bases, and we have an enzyme that's going to get ready to split that open. It's not going to copy the whole thing. It has a starting place, okay? And this would be the start of where a gene is. The gene is the valuable information in DNA that we want to copy to make proteins. So we can just put here, I can just put start of the gene, okay? And that is called RNA polymerase because it's going to be making RNA. That's another important term. Okay, and this whole thing, okay, this whole process, like this is called DNA replication, this whole thing is called transcription. Out of transcription, that's where we're making the RNA eventually be made into proteins, for the most part, not always. What's some RNA that's never made into protein?
Is that the junk DNA? No.
Well, that's not RNA, it's DNA.
That's
It's
DNA.
not used.
Right.
But RNA that is made from a gene, can you think of an example that's not made into protein?
Just what goes into, no.
What organelle?
The ribosome?
Ribosome.
Is it with the
Right.
70% of the ribosome?
The ribosome is about 70% RNA,
Right.
and it's not made into protein. It's actually the structure. That's why you're copying the DNA to make ribosomes.
So the RNA that enters the ribosome is not necessarily copied into DNA or into a protein?
No, it is the copier. The RNA is part of the enzyme.
Okay.
Okay? So another one would be the transfer RNAs. That's not made into protein either.
Okay.
And we'll go over that process in a second. One very important thing, there are factors here, and it could be 3, it could be a dozen, it depends on what the cell needs to start that gene to be copied, okay? These are called transcription factors, okay? And very important. Transcription factors, and I'm going to put switches in parentheses because that's essentially what they are. These are the switches that are going to turn the gene off and on and let that RNA polymerase work. If you don't have all your factors in a row, that thing can't get started. And conceptually, 5 years from now, you're not going to remember these terms probably, but conceptually, you can be amazed at what they're doing because this is what tells a nerve cell to be a nerve cell. This is the transcription factors are what tell a nerve cell not to make digestive enzymes, okay, that would kill the other nerve cells around it, okay? So this is how you turn the genes off and on, is the transcription factor. And very often, it's another gene down here that has to be turned on by some transcription factors that will be the key. Its RNA will be coded into protein, and then that protein will be a transcription factor for another gene, okay? So you've got a cascade of these genes, and this is how you make arms and legs and eyes and things like that. You've got a gene that's a switch, it turns on, and basically that is going to turn on other genes, which possibly turn on other genes, and you get this cascading effect. But they all have transcription factors at some point that have to be there, okay? And so this is why kidney cells are kidney cells and that sort of thing. This is why your bones only grow as far as they do, because the genes are turned off, okay? If they don't get turned off, you grow long bones, you grow long fingers, okay? There's all sorts of things that will happen. If the gene for apoptosis that triggers the lysosome to kill the cell, okay, it's triggered cell death. If you don't have that, you'll be born with webbed fingers, okay? because in the embryo, the cells between the fingers have to die, okay? Otherwise, you're born with flippers, okay? But that's exactly what's happening. That's what happened when those people are born with that. The gene didn't turn on. So something happened in the cascade, okay? So that's the concept that is just really amazing. And it's really what makes humans human, okay? 98% of our DNA is just like chimpanzees. Even the mutations are similar. So we know we're related way back, okay? But you turn off a few of them, or turn on a few of them, and you get us, okay? You turn off the ones that make hairy bodies, okay? By mistake, you can turn it back on, and I'm sure you've seen pictures of, like, the wolf boy, you know? That's what's happening. They're turning on those genes again, and you're getting full body fur, okay? So we're mutations just like chimpanzees have mutations, okay? And that's what makes us us. It's this kind of stuff, okay? So, all right. So at the next stage of this same process, we're still in the nucleus, okay? This is where the RNA polymerase is going to be reading the bases. It has to separate the DNA first, okay? And it's going to pick the one side that it wants to read. So this could be an A, a C, and a G, okay? That's one, and that's called a codon when it's in the RNA. Sorry, we're still in the DNA here, okay? But this RNA polymerase has been copying these things, and now there's a strand of RNA, okay, that has 3 base pairs, and every 3 of those is called a codon. So it's just like the triplet in the DNA, only in RNA we call it a codon. Okay?
Can I ask you a question?
Sure.
On the DNA, you've got the T and the A and the C and the G, but in the RNA, you've got a U replaces a T.
Right.
What's the difference between, or does that not matter for us right now?
It really doesn't matter for you, and it's not something I would test you on. But that happens. You're right. You switch the T for uracil, which is a very similar base.
Okay.
So it's just a little different structure, a different little amine on there. So, but basically, it's the same kind of thing. Okay. Now this RNA here, let me label it for you here. RNA. Okay, I'm going to put an M there, and the M stands for messenger. Okay. The mRNA, this is what's going to be coded into a protein. Okay. And it's going to have to find its way out of one of those big nuclear pores that looks like a kitchen sink strainer. Okay? So that RNA is going to be snaking out of the nucleus. Okay? It will either land two places. It can land on the endoplasmic reticulum. Okay? There's the membrane, which has ribosomes stuck to it. So it's rough ER. Okay? Or they're just ribosomes out in the cytosol. Okay? And they're going to be reading RNA all over the place. Okay? So I remember seeing all those little red dots all over the cell. Okay? But some of them are stuck to the endoplasmic reticulum. These, the red here, tend to be proteins that are going to be put in vesicles. Okay? Whereas these are reading proteins, making proteins that are just going to be enzymes out in the cell. Okay, like globular enzymes and things like that.
Those are the strands that just go through the nuclear pores, the nucleus pores?
The RNA does. But after the RNA gets out, the ribosomes aren't in the nucleus. Okay,
it's in the cell.
It has to get out
Okay.
to get on the ER or out in the cytosol to be read in the proteins. Okay? Let's see here. This. Let's put these over here. Okay. Or a ribosome. Obviously, that's an important term. Okay, so let me label that for your word bank there. Nuclear pore. Okay. Double lipid membrane, nuclear membrane. That's kind of a nice fact to keep in your head. Okay, I'll draw 2 layers here. There we go. All right. Now the RNA is out here. I'm going to draw a big ribosome around it here. Okay? This is a short piece of RNA. Somewhere in the center of the ribosome is the enzymatic portion where the other form of RNA, the transfer RNA, is basically RNA that's folded up on itself like this, and on one end it has an amino acid attached to it. But this part right here, it's still a strand of RNA, so it has bases that are sticking out. Okay? And the three here, these are the bases right here that have to match the codon on the messenger RNA. So that's why the codon specifies which amino acid is going to get put on the next in the chain. Okay?
So it's the three bases of the transfer RNA matching the three bases on the messenger RNA
Right here.
that establish what amino acid gets kicked off.
Right. Let's say there's these three, and here's the messenger RNA, or the transfer RNA,
Okay.
with an amino acid right here.
Okay.
Then one comes in next to it. There's the RNA, it's the transfer RNA, with another amino acid. But it's the code, it's these three code that codes for this amino acid, this three that codes for that amino acid, and they eventually get hooked together.
To form the
Okay?
protein chain.
And that's the protein. And so the protein is going to be growing out one side of the ribosome, while the RNA is pulled through like a ticker tape on the other side. Okay?
Okay.
But it's the transfer RNA, let me write that. You can call it tRNA for transfer. And I'll just put parentheses there. And this is actually called the anticodon because it matches the codon on here, but don't worry about that. Just to remember codon and triplet forming, that'll be good. Okay. And this whole process, let's put the word up. Okay. Translation.
Translation.
So it's actually like a book. The information is transcribed, okay? Then it's sent out into the world and translated into different languages, okay? Think of it that way, maybe. And this is how all proteins are made. So this is the ribosome is the key, okay? Mostly RNA. Let's put that up there. Right ribosome. Okay, ribosome. So translation. That term, transcription, replication. Don't walk into the lecture test not knowing what those mean, okay? That's definitely going to get some questions. How many, anybody remember how many codon possibilities we have?
64.
64, that's right. 64, right. Since there's 4 bases,
Yeah.
we can match it up 4 to 4, okay? Gives you 16. And then times 4 again is 64. Okay? So that's how you get those. All right, question? Well, you're matching up 4 things in 3 different ways. So you have to go 4 times 4 times 4. And that gives you 64.
And then I should be called 64 amino acid possibilities.
Yeah, but we don't even need 64. We only have
We need
20,
20.
right?
But we can make, there's a
Well, life
possibility.
just doesn't do that. You know, it only uses the 4 to 20 amino acids. All
Okay.
life uses pretty much the same amino acids.
Okay.
And so you have duplicates. Some of the codons are different, but they'll code for the same amino acid.
Okay.
So some of them, like leucine, have like 4 to 5, 6 codons that actually code for that one amino acid.
Because of, we showed us a chart where
Yeah,
if a U hits, or if a T hits an A instead of a C, it's okay because it's still in the same general area.
Sometimes,
Sometimes,
yeah.
and then other times it's not.
Right.
Okay.
That's when you have neutral mutations. It's going from one thing to another, but if it doesn't affect the amino acid, it's neutral.
Okay.
It hasn't changed anything. So
Okay.
I think that's pretty much kind of the key words. We'll go through the slides real quick and see if we find some others. But if you can, sort of understand these things and know what they are, know what they're doing, know what the ribosome is doing, one little fact from way back, remember? What part of the nucleus makes the ribosomes?
Nucleolus.
All right. Any other questions about this issue? So, I'm going to go through both sets of slides, and we'll make it with plenty of time, I think. And I'm just going to hit the highlights here. Just a picture of the cell. And also, don't worry about the lipo. Okay. I'm just going to skip right to the dot here, right here. Okay. Basically, I think we should know a one-line definition of each of these terms. Okay? Because I'll do some sort of true-false or something with them and blow your mind. All right? So, the spontaneous generation, basically life from non-living matter, like maggots from meat or mice from straw, things like that, the old theory. The miasma theory was the bad air. Right? Malaria. Okay? Cell theory is basically the theory that life involves cells. Okay? And that you cannot do anything that your cells can't do. Your cells don't have the genes for making neurotoxin snake venom, okay? So you can't bite people and kill them like that, okay? But snakes can. It depends on what the cells can do, what kind of genes they have, and all life sort of breaks down to the cellular function. And most disease does too. Germ theory, the idea that microbes can cause disease, invisible little beasties, which was very hard to accept for a long time for people, okay? But that's basically what germ theory is. And vitalism was the idea that there were special kinds of compounds, like urea, that only living things could make, that you couldn't make them out of inorganic matter. Okay? And just so you know, they've actually created, you could call it artificial life. Greg Winter out in California was the first guy to do the human genome, but he's sort of an innovative guy, multi-millionaire scientist who got together a lot of brains, and basically they synthesized DNA from scratch. They took phosphate, they took ribose, and took the bases, and they synthesized all the genes, about 400 genes, I think they figured that was basic for living. They looked at all the bacteria and looked at all the genes that they all had. In other words, the very basic set that you have to have for life, not to be a special pneumococcus or streptococcus, but just sort of special life. And they said, this is about the basis of what we have to make. And they made them all artificially, took the nucleus out of a bacterial cell, put theirs in, and it's now living. It's artificial life, basically. The genes, the DNA was made artificially. And it has now, it's reproducing, and now that they've fed it, it has made all the proteins that it needs, and now all the other stuff in the cell is made from artificial stuff too now, okay? Because the old stuff was either eaten or worn away or whatever. So it all has new ribosomes, it has new lysosomes, and all that sort of stuff. So it's basically an artificial cell that's living, okay? And this was just done a little over a year ago.
Wow.
So you can see what kind of fantastic stuff is going on now in the genetics world.
What would something like that become, or would it
It's
just
bacteria.
just bacteria?
It's harmless.
That's
They didn't want to give it any kind of virulence. Okay?
Okay.
So they made sure they didn't give it any genes that would let it bond to human cells and things like that.
Okay.
So,
That's nice. A couple of years when I was
Yeah.
but now, what they're working on is taking bacteria that can eat things like oil, okay, and degrade it so that you can let this stuff grow in oil spills and it will just get rid of the oil. It'll eat it and it'll be carbon dioxide, okay. They're also developing bacteria, and this is already going on, that can make hydrocarbons from normal food sources like cellulose, grass, things like that. The bacteria actually secrete hydrocarbons like gasoline, okay? And you just separate it out, and they're looking at artificial fuel sources like this. So, and if you think that there's no way you could make a lot of gas that way, just think of how much beer is made and wine is made in this world, and all the bacteria and fungi and everything that are responsible for making that stuff, you know? So you can make a lot of it. Anyway, okay. Now, PBS moment. But fascinating stuff. Okay. So, but anyway, vitalism was the idea that life had that special essence, okay, that you couldn't make it artificially. This was just to sort of show you this, and just sort of remember the term fluid mosaic model and what it means. There's your word bank there. Okay. It's a phospholipid bilayer, or a lipid bilayer, however you want to describe it that way, with proteins and things in it like cholesterol and sugars on proteins and things like that. They're all sort of floating around in this membrane. They're not really constrained necessarily. But that's called the fluid mosaic model. Okay? Remember the name integral protein, okay, which is sort of the same as transmembrane or membrane proteins. Okay. All right. This was just, you don't have to know the specifics about the sodium potassium pump. It was just an example of a membrane protein, okay, but it's also a big example of active transport, okay, because it takes that ATP to pump out the sodium and bring in the potassium. The gastric acid pump, again, was just an example of a membrane protein. You don't have to know anything special about it. Okay. Just know what an aquaporin is. Okay, it's a pore that lets water into the cell. Why is that important?
You need the oxygen
To keep
from
all
the
the
water.
stasis,
The
to keep
oxygen.
the balance going, and either too
balance
much
of
water
what?
of water.
No,
Okay,
it's
what
what
do we call
pushes the
that?
hydrogen out.
Oh, what's that word?
Starts with a big O.
Isotonic, hypertonic tenacity.
Osmosis.
Osmosis, yeah.
Osmosis. If you don't have the water for it,
Yeah.
there is no osmosis, because the water can't get through that lipid bilayer. Okay, so you have to have that. So just know what it is, basically. Okay. And know that all life has to have it, because all life has to have osmosis, right? So even bacteria are going to have pores. I think it's just kind of interesting that we have alpha helixes in our pores, and most animals do, okay? But mitochondria have bacterial beta sheets in their pores, okay? Which is just another indication that mitochondria are bacterial in work. So, potassium channels, again, this was just an example of a membrane protein, no specifics. This happens to be one you're going to get tired of hearing about in the next week, because we're going to start talking about excitable membranes and ions moving through membranes and things like that. Nerve conduction, that's exactly what it is. Potassium and sodium going through the membrane. So, again, just an example of a transmembrane protein. Okay. Same here on this part.
Do you want us to know that it's got the 4.5 pH in that?
Not specifics, you know,
Okay.
but you shouldn't remember that you have to have that acidic environment for the mitochondria to work.
Right, because that's what then forces the hydrogen back in, which takes the ADP into the ATP.
You got it. That's the only reason we eat, is to get this pump working, is to get the
Right.
acid out here so that this pump can work.
Okay.
That's why we eat, basically. Okay? Other than just building proteins and building bodies, to actually live, though, this is why you eat. Okay? And otherwise, like I said, this was just an example of membrane proteins. And everybody remembers the other organelle that uses acid, right?
Lysosome.
Lysosomes, the digestion organelle. That was just a shot of protein, PBS. What do you need to know about the nuclear? Well, the nucleus has an organelle. Well, that's where your DNA is stored, for one thing. That's where the nucleolus makes ribosomal RNA. Okay? It is a double lipid bilayer. Okay? It's got two bilayers with large pores there that sort of look like that kitchen strainer I was telling you about in the sink. Okay? So just those few facts, I think is...
That's the nucleus or the nucleolus?
That's the nucleus.
That's the nucleus.
That's the pore. Those are the pores in the nucleus right here. Okay.
But isn't it the nucleolus that's what's making the RNA for the ribosomes?
Yeah, that's in here.
Right, right,
Whole
but
thing is
That's,
Yep.
that's where the nucleolus is making that special RNA that
Okay.
becomes a ribosome. But it still has to get outside the pore to become
Right.
a ribosome.
okay.
Okay? So, but the rest of this chromatin over here is being made into messenger RNA, and that's going out the pore, and that's going to be made by the ribosomes into protein.
Okay.
Or decoded by the ribosomes into protein. So,
more sense.
What else, little tidbits?
I remember you said something, I just wrote it down, it said that it folds into the,
Endoplasmic reticulum,
Yeah.
right here, that's right. I
Okay.
think that's kind of a cool fact.
Yeah.
Okay. The outside membrane there just folds back on itself and actually becomes the endoplasmic reticulum.
That's interesting that it uses that up to make another organelle.
Right, right. And I didn't know that until just a few years ago. Because I always wondered,
What
you know,
happens?
when we were studying mitosis, what happened to the nucleus?
Right.
What happened to the membrane? And so I guess I just wasn't curious enough to really pursue it at the time. But, here's what it looks like. That just happens to be some pathology. Extra credit if anything, okay? I would know the difference between simple diffusion and facilitated diffusion and know that they're both passive
Passive.
transport mechanisms. Simple diffusion is when something like, say, oxygen or carbon dioxide or something diffuses across the membrane. Okay? And it's also when a pore, that could be a water molecule, okay? And it could be going through a pore, that's still simple diffusion. We call it osmosis, but it's still simple diffusion when water moves through the membrane. Okay? Over here, this is a protein, it's not active, it's not using ATP, but it might have sort of a receptor site that only takes certain kinds of molecules, like starches or something. And once they land in that thing, the protein changes shape and sort of dumps it out the other side. But the only reason it does that is because that molecule is flowing down its concentration gradient. Okay, it wouldn't go in if there was more stuff inside the cell than out. Okay, so that's still diffusion, but it's facilitated. Okay? Because most molecules won't diffuse through the membrane, the lipid bilayer. Okay, sugars are not going to diffuse through. They're not going to go through with all those hydroxyl groups. They're not going to go through the oily tails of those membranes. So you have to have some method for them to get in. So. Then we talked about active transport. This is probably one of the more important ones. The sodium-potassium one. Like I say, don't worry about numbers just yet. But just realize that it takes ATP to pump that potassium in against its concentration gradient. Okay? Because there's more potassium in here, it wants to go out. So that's why you need the ATP to pump it in. Okay? Same way with sodium. There's more out here than there is inside. So you're pumping the sodium against its concentration gradient. Okay? So it wants to go inside, and that's a nice fact because now we can use that to drive other paddle wheels here. We can use that sodium gradient out here. It wants to get inside, and we can use it to drive a protein that will drive, say, a calcium out. Okay? So we're not using ATP here, but this one is dependent on this one to work. That's the whole point of secondary active transport.
That's
So if that's,
indirectly.
because I wrote no pump, no transport. So if the ATP pump up top is not working,
Right.
then the bottom one can't.
Can't work. There would be no
Indirectly
concentration
dependent.
gradient. Okay, everything would just, once it diffused into almost, or equilibrium is a better term, nothing's going to move. And this was just some other examples. You remember what you call the ones where you have two things moving in the same direction?
Symport,
Symport.
Symport, good, good. And against each other,
antiport.
antiport, right? Right. Oh, okay, there you go. Symport and antiport. Maybe one question on that. Okay. This was just to show you examples of these things. Don't worry about the details. Okay? Everybody should be familiar with most of these terms, so I'll just pass on here. Notice. Nope.
Makes the peroxisomes.
Well, this is the endoplasmic reticulum. Okay. These look like they got crista there, so these are probably mitochondria. And you've obviously got the nucleus and the nucleolus. But this is a much more realistic picture of a cell, especially a cell that makes lots of proteins. Something like a pancreatic cell that's making lots of digestive enzymes. It's got to have a lot of rough endoplasmic reticulum to make all those proteins and keep making them. Okay? So these are all little ribosomes here on the endoplasmic reticulum. All these little bumpy areas, those are all ribosomes. So every one of those is turning out proteins all the time. Okay? So that's kind of a fact. that I think everybody should know by now. Okay, the rough endoplasmic reticulum tends to make proteins. Okay. Other than that, there is some storage, like of steroids and glycogen, inside the membranes. Okay, because eventually these things, those membranes are going to fold out into vesicles and send them to the Golgi, right? And so it stores some of this stuff. But I wouldn't get too much more specific than that.
You had told us before that the ER makes the peroxisomes.
Yes, it does.
Okay.
Yeah, but that it forms a
Right.
vesicle.
Okay.
With those oxidating enzymes inside in the
Okay.
membrane.
Okay.
And occasionally, well, much more
Okay.
than that. And this just sort of illustrates that pathway. Don't worry about the terms or anything like that, but the endoplasmic reticulum is making vesicles. One of these could be a peroxisome, okay? But otherwise, they're going to fuse with the Golgi apparatus. They'll be modified and then made into other things like lysosomes or secretory vesicles that are going to go up to the surface and dump the enzymes outside, okay? So that's what the Golgi does. It modifies things that came from the endoplasmic reticulum. And I think that's clear enough on there. Again, this is redundant, just what I just said. It just sort of gives you that picture. Don't worry about all the writing and everything. Lysosomes. What kind of facts, important facts, come to mind when you think of lysosomes?
Digestive enzymes.
Digestive enzymes, and it's an acid pH. Here, if you remember 4.5, but it's an acid pH, and these are called acid hydrolases. Okay? And those are the enzymes. Other than that, you're good to go. And this is just an example when it goes bad. And those, there's all sorts of. You can see that the lysosomes do a lot of different things. They have dozens of digestive enzymes, and all you have to do is knock out one of them, and you're going to have one of these diseases. Okay? You're going to have a storage disease. If you can't break down something, it's going to build up, and you're eventually probably going to have big problems with it. Okay, I think everybody by now remembers what the mitochondria looks like. Okay, that ATPase is inside this membrane right here. The acid is being put out into this space out here, and it's going to rush back in to where the blue area is, and that's going to turn the wheel. There are special pores and receptors for getting the ATP out through both of these membranes. Okay, in case you're wondering how the hell does it get out? Okay. There are special pores and receptors for it. And likewise, receptors to bring in things like sugars and the pyruvate that's being used in the Krebs cycle that's generating those hydrogens, okay, the oxidative part. So, I think it's nice to remember that mitochondria come only from the mother, okay, and this is why you can trace your maternal origins, okay, by just using the mitochondrial DNA, okay? Because we keep sort of neglecting the mitochondria. When we talk about our DNA, well, mitochondria have their own DNA, okay? They make a lot of their own enzymes and proteins and things, and they have their own ribosomes for making proteins and things like that, so it's bizarre, okay? And I think, I may have just misspoke. I want to double check on that. But they are basically only from your mother. Okay? Bunch of different problems. All those wheels turning and all that stuff in those membranes, all those integral proteins. You make one of them not work, you're going to make the whole mitochondrial process malfunction. Okay? If it's fatal, it's fatal. If it's not quite fatal, you get diseases like this. Okay? That's the only thing you should have in your head for that.
Just saw on the TV like 3 days ago about Wilson's disease. They had like a half-hour little thing on Wilson's disease.
Yeah.
It was really interesting to relate it and understand why they had the problems they have.
Well, almost every one of these diseases has websites. where patients who have the disease go and give
Get
information,
information.
find doctors and things like that. So if you're interested, you can look up any of this stuff and see videos on YouTube. The patients video themselves showing their weakness and things like that. And so yeah, the internet is just going crazy with medical issues. You know, and, not literally crazy, but I think it's good. You know.
It's a good side of it.
Yeah, it's the good side of it. Except for the fraud who are trying to take advantage of these people.
They did a report today, though, about people trying to diagnose their own problems with Dr. Google there.
Dr. Google, yeah.
Yeah.
Well, you'd be surprised how often it works. Because you're motivated to find answers, and sometimes you look a little harder than your doctor does, or he blows something off. He doesn't think it's too important, and you're more concerned than he is, you know, that kind of thing. So I'm not saying it never works.
Yeah.
Okay, I've had doctors tell me they were surprised when a patient came in with something, telling them what they had, a disease that was fairly rare. Okay, and they admitted it. I probably wouldn't have thought of that. So, because you know, doctors, most of the time, you know, you hear hoofbeats, you think of horses, but occasionally there's a zebra running by. And a lot of doctors will miss the zebra because they're too used to thinking about what's always happening. You know?
Standard,
I'll give you one quick story. I had a fellow who came to see me, looked like he had the flu, low-grade fever, kind of congested, but he was just complaining about fatigue, you know, weakness. It was flu season, you know. He went to another doctor. The guy said, "You got the flu," and like every one of my patients today had, you know. But the guy was just, he wasn't satisfied with that. He just didn't feel like it was the flu. And he came to me, and his only complaint was flu-like symptoms, okay? But he said the other guy didn't do anything, so that's why I'm here for you, okay? So you can't really blame the other guy. You're not going to test everybody that comes into your office with the flu and do blood work and x-rays and all sorts of stuff. But I said, okay, I want to, you know, just satisfy. I thought he had the flu too. But you try to satisfy patients in their anxiety. And so I ran some blood work and got a chest x-ray to see if there was a little early pneumonia or something going on. They called me immediately back from the blood bank saying he had leukemia. He had a erythroblastic anemia. You know, he was weak, his blood count was way down, all sorts of blast cells in his blood that were abnormal. And they said he needs to go to Tampa to, you know, right now, okay? And so we transfused him, I think on the way, because he was so anemic, but he died 2 weeks later.
Wow.
So, you know,
Yeah.
what if I had done nothing?
Right.
Sure. So that's why you take people seriously, especially if they're anxious. You got to satisfy what they want, even if you don't think it's necessary. Okay. So, and, well, anyway, that's just one story. There's a lot of them. Okay. All right, peroxisomes. When you think peroxisomes, you think oxidation. Okay.
Detoxifiers.
There are several enzymes that do this. The big one is catalase. Okay, so that's an important enzyme, especially for breaking down things like alcohol. And it oxidizes the alcohol to aldehydes and things like that. But the peroxisomes are also important for oxidizing long-chain fatty acids into shorter-chain fatty acids, which can then be broken down and burned as fuel. Okay, and when you can't do that, you get other diseases, like the one coming up. But it sounds kind of complicated, but associate peroxisomes with catalase and hydrogen peroxide, okay, because that's one of the oxidative factors. And then think about that and peroxisomes and alcohol, because that's what detoxifies a fair amount of the alcohol you drink. Okay, so just those little facts I might ask a question about. Okay, and there was one where peroxisomes don't work. Okay, so there's centrioles. Not too much to know about centrioles because there's not a lot known about them other than the certain proteins that are in them that function, but we still don't know exactly what turns this stuff on and how these proteins are working and what triggers it and how they generate the microtubules and things like that. I read a paper a while back, they were talking about, you know, just dozens of proteins having to get together to function almost like an RNA polymerase, okay, to make the microtubules to get them started. Okay. But so just associate centrioles with microtubules And you associate microtubules with what? The spindle apparatus in mitosis, and flagella and cilia.
Yeah.
And that's what makes them move. Okay.
Can I have a question for you?
Yeah.
You had said this before, at least I think this is what you had said, the microtubules form the basis of the cilia and the flagella. And then it's actually the motor proteins then
Right.
that make it move. So is cilia a microtubule, or is it kind of like,
It's many. There are several.
Okay.
It sort of looks like this. It almost looks like a microtubule barrel.
Okay.
But those microtubules are rubbing against each other because of the motor proteins.
Proteins.
That's what gets it to bend in its shape.
okay,
That's what gets it to move.
the cilia actually is microtubules?
Yeah, it's sort of made of
Okay.
but it's got a membrane around it.
Okay,
Okay, so it's not just like hair growing out of your skin.
okay,
It actually has a cell membrane around it.
okay,
But they're still flopping.
okay.
Okay. And ribosomes. Well, I think we all got a fairly good idea what ribosomes are now, right? There's all that RNA. The blue part is the protein, and this is actually the small section. This is what they call a 30S subunit. There's a bigger unit that has to grab onto the other side of the RNA before this thing works. Okay? But just remembering that they make protein, they're mostly RNA. They need transfer RNA, though, to bring those amino acids to them. Okay? And then they do their job. So that's the last slide in the first part.
Please bring the transfer protein to bring the amino acids to them. Okay.
All right. I'm not going to get too specific with questions about this part right here. What's important about cell adhesion? Well, if you don't have the right proteins, if you can't do it, you can't make multicellular structures or animals. You're still a bacteria if you can't get together with another cell and communicate and do things. Okay. And these are the methods that life has come up with for hooking these things together. Different types of proteins. These are all, evolving at different times and different rates and that sort of thing. Different types of cells use some of these more than others. But basically it's a way to stick together. Okay. So, but otherwise we're not going to get too in-depth on the questions. Okay. Now the gap junctions, I think the important part here is realizing that that's what they look like. Well, not really, but I'll show you what they look like. But they're hollow, so ions and things and small molecules can get back and forth between the cells. And this is really important in cardiac muscle and smooth muscle. If you want a regulated movement, if you want a rhythmic movement, okay? Think about it. Your heart has, billions of cells, muscle cells. If you actually have a nerve that stimulated each cell, like you do in your skeletal muscles, okay, you'd have to have a huge nerve all around your heart, okay? But that's not how it works. You have, certain cells within the heart that are well, I can tell you how this when we get into the membranes, but it fires, they fire themselves, okay? But that trigger, calcium, is the trigger, and it's going through the gap junctions, causing the next cell to fire, and the next cell to fire. So you get this rhythmic wave of contraction, which gives you the beat, okay? And it's the same thing that's happening in your intestines, okay? Smooth muscle is doing peristalsis and moving things through, okay? And the smooth muscle in your esophagus is doing the same thing, squeezing food down, right? There's nervous control that dumps things like adrenaline on these cells and can speed them up or slow them down. But they have their own regulatory movement. Okay.
Can I ask you one quick question about that? It kind of has to do with it, but pregnant women have to have a lot of calcium, obviously, so the baby has strong bones and teeth.
Yeah.
But is it also because of the gap proteins and the body need more calcium for that contraction to happen when they deliver?
No, not really.
No?
I mean, you'd have to have a huge uterus to really need a lot more calcium.
Okay.
The,
Not the vision I was looking for, but okay.
and you've got plenty of calcium in your own bones.
Okay.
You know, your body is always taking calcium from your bones when it needs it,
Okay.
and then putting it back when you don't need it. So depending on your diet, you're doing one or the other.
Okay.
So that's regulated by a lot of the kidney hormones.
So basically, smooth muscles rely on cell adhesion or gap junctions.
Well, both. I mean,
Okay.
they have to stick together.
Okay,
Okay, but the gap junctions are kind of special for them.
okay.
Okay. Vital, I should say.
Mhm.
I don't worry too much about adhesion belts, but I think just knowing their structure is fascinating. Okay, this Velcro kind of proteins here. Okay. And then tight junction, these are just like rivets holding these things together. Gives you a watertight seal around the epithelial linings. Okay. Here's an example in an endothelial cell, tight junctions from head to tail here, pushing this thing together so fluid inside here can't leak out between the head and the tail. And so that's waterproof now because of that. It's fascinating to see all the endocytosis that's going on here with these vesicles. They're being transported over, and then exocytosis gives the stuff to the cells that live on the outside. Okay. Just another picture of gap junctions. Okay. That's what they really look like in the electron microscope. You can actually see the holes. Desmosomes are important for holding cells together like this, especially epithelial cells. And if you take half of a desmosome and think of this protein right here grabbing a basement membrane of other proteins, that's how the cells stick to the basement membrane. Okay, they stick to each other like this, but they stick to the basement membrane with sort of half of that structure. So we wouldn't have evolved multicellular animals if we didn't have these things. And this was just an example of when the one that stuck to the basement membrane, when it goes bad, your epithelium lifts off and forms blisters. And that's all I was trying to illustrate here. All right, we've already talked about that. I think just a basic fact that clathrin exists, just be aware of it, and you have clathrin-coated vesicles. The clathrin, because of the way it's structured, when it crystallizes, it forces the membrane into a circular form. Okay, because of, like, they look like this, but the clathrin is actually a sort of a three-legged thing, and when they crystallize together like this, they form this sort of structure which forces the membrane into a circular structure. Okay, so that's how some clathrin vesicles are formed. Okay, just if you know that, you're fine. And then we just went over this earlier. Let's see if there's other words I want you to know. Okay, we went over chromatin and histones and nucleosome. We went over triplets, what they are. We went over the term replication, anyway, okay, and how it's made. We talked about helicase being responsible for tearing open the DNA. It goes bad. DNA polymerase, blah, blah. We talked about some ways that you can artificially mutate the DNA. That's mostly PBS. Know the definition of an exon and an intron. Okay. Exon is the good part of the gene. The intron is the useless part that has to be clipped out. Am I going too fast?
You said the exon's the good part, and the intron is the bad part that gets clipped out?
Yep. Where do you want to go back to?
What the hell? Just started looking right now.
Well, the DNA was not too far back. Right here. Okay. Or I guess this is important too. I mean, like I say, don't walk in that test not knowing what transcription means and what translation means. Okay? We get a couple questions on those. All right. That's what DNA looks like. It's called a double helix. I think that's a good word to have in your word bank. Double helix.
It's not a double-stranded protein? I'm just joking. I'm just joking. I'm just joking.
I almost had a stroke. Okay. Chromatin, histones. Basically, just know what they are. Okay. Okay. DNA coils into chromosomes. That's basically all that was shown. It's chromatin, and when it coils up, it's chromosomes. Okay? And that's what will hold that stain. That's why they're called chromosomes. Anytime you hear the word some in medicine or biology, soma means body. So it's like chromosome means colored body. Okay? That's because when they dye it, it's stained. That's why you saw them. Okay? So the nucleosome, okay, it's the body in the nucleus. Okay.
That makes sense.
Okay. Talk about helicase being the first part of the DNA. Yeah, not much here you need to know. So we're starting over here again. Exons are the useful part of a gene. The introns are the unuseful part that needs to be flipped out. Okay? So that this part of the exon here will be hooked to this exon afterwards when the spliceosome is done. Okay? And I don't know if I explained that this is how you can get hundreds of thousands of proteins out of just a few genes. Because depending on, most of your genes have to be spliced perfectly. The intron has to be taken out just right. So that, and what happens is the spliceosome folds it up. So then it connects the two good pieces together and clips off the loop. Okay? So now you've got a good piece of RNA that's going to go be read into a protein. Okay? What your immune system does is it randomly splices. Okay? So now you get all sorts of weird connections of proteins that fold up in weird ways that just may happen to have the right structure to latch onto things. That's what antibodies do. That's how you make them. But that's how 23,000 genes in your body can make hundreds of thousands of antibodies. Okay? So it is almost an infinite number that can be made. Just by randomly splicing out certain sections, you get a brand new protein every time. Okay? So, don't have a stroke over this. I think if you just sort of realize that words like deletions and insertions and gene duplications, these things are abnormal sort of, mutations. Okay? Maybe one question about this. Epigenetics, remember what that means? It's the changes to the DNA structures and the histones and things like that don't involve any sort of real mutation of the DNA. Okay? So you're changing gene expression
Because you're
without
messing.
actually changing the DNA.
Because you're messing with the histones.
The histones, yeah.
Okay.
Usually.
So, can you repeat what you just said?
Yeah.
That it's
Well, on or above, epi meaning on or above.
right.
Okay, so this is sort of above the DNA code.
Okay.
It's the next step. You can change what the DNA code is going to do
Okay.
by screwing around with histones.
Above the
Yeah,
DNA.
so that's above the DNA. You're controlling what's being expressed.
Okay.
Your genome may want to express some sort of gene, but if you've been exposed, say, to some toxic chemical that has screwed the histone on that particular gene, you're not going to express that gene.
Okay.
Okay, at least in that cell.
Okay.
Okay.
So it's basically, I'm paraphrasing here. It's basically gene mutation, but
Artificial.
because
Yeah.
you're not going through the DNA
You're not changing the
directly.
DNA. Right.
Okay.
This is why twins don't have the exact gene expression.
Okay.
Okay, they have changes to their histones. You can even look at the chromosomes. They look alike at birth. Okay, but as they get older, the chromosomes start to look different. It's because certain genes are turned off and others aren't working and things like that. So even twins with identical DNA have a different functioning group of genes.
Okay.
Okay, and this was just sort of illustrating transcription, which is almost the exact thing I tried to put on the board. Okay, you have the RNA polymerase that's going to read the gene. You have the transcription factors or switches here. Okay, and then when this whole thing gets started, it looks like this. Okay, the RNA is reading this part of the DNA and making the coded pairs here. So this is the messenger RNA that's coming off. This is what's going to have to go outside the nucleus to be made into protein by the ribosomes. Okay. So that's why they call it the messenger RNA. Okay. And as soon as you hit the stop code, one of those three bases that codes for a stop, the gene is over and the polymerase will leave. Okay. But if you need to keep making that kind of proteins, you're just going to have more RNA polymerase hit the same gene and keep reading. Okay? So you're making multiple copies of these proteins. This was the 64 possibility. Okay? That's all you need to remember. And ribosomes are mostly RNA, some protein. They take two halves to work here, but they make the protein with the transfer RNA delivering the amino acids according to the code. Okay? Just those basic facts is all I'm going to ask about. Okay? And that was more PBS than anything. This is kind of what the picture I tried to draw as well. Okay? This is the ribosome. Here's the RNA that's being read, and here's the base pair that's being paired up. And as the amino acid is added onto the chain, then that transfer RNA will leave. Okay? Go pick up another one and come right back. Okay? Just by random diffusion. Okay? That's how they get in there. It takes a little time. I mean, this is not speed reading like the DNA polymerase that can zip down your DNA and do thousands probably per second. Okay? Because these things are big molecules, and you've got 64 of them floating around, 64 different types floating around. You could have 63 of them come in and say, oh, no, not right one. You know, and finally the last one matches the code with and then adds on the amino acids. So it's not a fast process. Okay? And then lastly, and we're done. We've got 10 minutes. Any questions at all? Break it.
So the RNA enters the ribosome with a messenger protein. So the RNA enters the ribosome with a messenger protein. Messenger protein brings messenger RNA. Okay. So the RNA enters the ribosome using a messenger RNA, and then a transfer RNA meets up with that, and if they're an exact fit, then it drops. Then it says, okay, you're going to get this amino acid, shoots it out in the chain, and then transfer goes back and gets more. Okay. And it just keeps happening as many times as it needs to.
As many times.